All right, welcome everyone. So today we're talking about epigenomics. So here we are in the third lecture of the second module. So basically the second module is about gene expression and epigenomics, and we're now entering epigenomics. On Thursday, we're going to be talking about three-dimensional chromatin interactions, and then we're going to sort of switch to module 3. So we're going to start with an introduction to epigenomics. Basically, what is the epigenome? Quick overview of the diversity of chromatin modifications, antibodies, how to carry out ChIP-Seq, and then a lot of the sort of primary data processing. How do we deal with epigenomic data? How do we map reads? How do we call peaks? And then higher level interpretation. So basically, this is a primary data processing. This is the sort of next level of interpretation. How do we discover and characterize chromatin states, which are getting a little closer to the biological function of the epigenome at that location. And then we're going to deal with several issues on model complexity. So a lot of people have asked, hey, how do we deal with how many clusters do we pick in k-means? How many states do we pick in an HMM? So now we're actually going to directly talk about how do we discover an optimal number of states, and how do we use some heuristics for actually optimizing the number of states, optimizing the selection of states at any one number of states. And then we're going to talk about how do we learn chromatin states jointly across many cell types. And lastly, about epigenome imputation. So first things first, let's talk about epigenomics. So the epigenome is largely credited with the ability of our genome to result in many different cell types. We have a single fixed genome, which is nearly identical in the vast majority of our cells. And yet we end up with this huge diversity of cell types. If you look at skin, there's this huge complexity of interactions between all these different types of cells, each of which is performing a distinct function. If you look at our heart, again, extreme complexity in the diversity of cell types. If you look at blood, there are dozens of blood cell types, each of which carries out distinct functions. And if you look at brain, again, there's extreme specialization in neurons, microglia, astrocytes, oligodendrocytes, which project and interact with each other, and so on and so forth. So how do we arrive at such an incredibly complex machine that makes a human function from a single genome? So the answer, in part, lies in the packaging of the DNA. Namely, there's about 2 meters worth of DNA in every one of trillions of our cells. How is that possible? This is possible through this 50,000-fold compression that happens through an extreme compaction of looping the DNA around these histone proteins, which form octamers known as nucleosomes that DNA is wrapped around. And then these are compacted into higher order structures, and higher order structures eventually leading to the chromosomes that we know and love. So this packaging has a role not only in condensing the genome and making it fit in every one of our cells, but also in selectively keeping regions accessible that will be useful to every one of our cell types according to its function. So depending on the specific function of every cell type, we end up with a particular packaging program, if you wish, for that cell type that then enables the functions of that cell type to be easily accessible. The analogy that I like to give is that every human on the planet inherits 23 volumes of all of human knowledge. But depending on your profession, you only use subsets of this encyclopedia, subsets of all of human knowledge, depending on whether you're a physicist or an artist or a gymnast or a computer scientist. You will use different aspects of that body of human knowledge. And the way that you do that is by bookmarking on that book of life, on that book of human knowledge, the pages and the passages in those pages that are the most important. Thank you. And the way that these bookmarks work is through epigenetic modification. So why are they known as epigenetic? Because they sit on top of the genome. Epi in Greek means on top. So genetic means, inherited. Epigenetic are things that are on top of inheritance. But epigenetic has a connotation of genetic. So very often you'll hear me use the word epigenomic, which has no such connotations. It simply says, this is the information that sits on top of the genome. Everybody with me so far? So what are the three types of modifications? So again, here's the chromosome, here's the chromatin fiber, here's the DNA itself, and the DNA itself can be modified by adding this CH3 group, this methyl group, directly on CpG dinucleotides. If that's a C, it's added on top of the CpG. And CpG methylation is usually associated with repression of regulatory regions. So when a regulatory region is repressed, it usually means that it's methylated. And that's one type of modification. The second one is the accessibility. So basically whether a transcription factor can actually bind in this particular location based on the positioning of the nucleosome. And then the third one is the tails of these histone proteins that make up nucleosomes. So every nucleosome is made out of an octamer of H2A, H2B, H3, H4, usually 2 copies each, that has long amino acid tails, and these long amino acid tails can actually be epigenomically modified by adding usually methylation groups, acetylation groups, and many other types of groups. Raise your hands if you're with me so far. Awesome, great. So this is like the basics of epigenomics. And now, this is fairly simple, adding a methyl group. This is fairly simple. Is it accessible or not? This is very, very complex. So histone modifications themselves have more than 100 different variations. So you can have, many different residues. You can have many different modifications in each residue. So the way that we denote them is by basically talking about histone H3, H4, H2A, or H2B, and then talking about the specific amino acid residue. So basically if the 4th amino acid is a lysine or the 36th amino acid is a lysine, and the specific chemical modification, is it a methylation, phosphorylation, ubiquitination, and so on and so forth. And then how many times? So basically if I add a methyl group on top of methyl group on top of methyl group, I end up with Me3, okay? And a shorthand we'll use is H3, K4, trimethylation, to basically denote 3 methyl groups adding on the 4th amino acid, which is a lysine, on histone H3, which makes up part of the nucleus. Yeah. Yeah, that's a very good point. So the question is, does the meaning of different marks change depending on where they are established? In other words, does the, could perhaps the same modification have a different function in different cell types? That is a very distinct possibility we worried about a lot. To all our evidence, it doesn't seem to be the case. At least the machinery seems to be recognizing the same marks in different cell types at the same type of function. So repression is usually methylation, H3K4 Me3 is usually promoter, regardless of the cell type. Very important question. Other questions? Which is important because basically if you mistakenly overexpress a protein that should be expressed in a different cell type that recognizes that mark, you don't want that mark to now start acting in an opposite way in that cell type. You always have to distinguish the cis-acting machinery, which is the stuff in the gene that you're looking at, and the trans-acting machinery, which is the stuff that comes from other places, the proteins that are encoded elsewhere. So the trans-acting environment does not modify the meaning of the cis-acting genomic marks. Other questions? Great. So again, we talked about this. And then the way to think about this is this constant struggle of gene regulation. Basically, transcription factors are fighting to occupy the chromatin, and then histones are pushing back. And then these general factors, chromatin compaction and all of that, is constantly competing to access the DNA. And every time one of them lets loose, the other one sort of takes over and so on and so forth. So it's this constant back and forth between these different processes. Okay. So these marks, as I mentioned, are extremely diverse, but they don't act in isolation. They act in combination. So if I have H3K27 acetylation alone, it means a different thing than if I have it jointly with other marks. So for example, in embryonic stem cells, if you have H3K27 trimethylation alone, it's a mark of repression. If you have H3K4ME3 alone, it's a mark of activation. But if you have both of them together, it's actually a different state known as a bivalent state that can actually go either way. So you have these two marks competing with each other. And if you have DNA accessibility alone, it doesn't tell you much. But if you have it in conjunction with other marks, then it sort of tells you, oh, this is an enhancer, and this is a promoter, and so on and so forth. So because of this potentially combinatorial role of multiple marks acting together, we basically need to develop methods for actually learning about which combinations of marks are actually meaningful and what is their meaning. And we'll see methods for doing that, and that's what chromatin states will be. So we'll basically develop a hidden Markov model, an HMM, whose hidden states will basically correspond to the true biological function of chromatin, which is never observed. What you observe are the emissions, which are the individual histone modification marks and the combinations of marks. So basically, we'll use these, HMM states to basically denote the true hidden state of the chromatin. Okay? Everybody with me? Yes. We're going to look at a few dozen. So enhancer, promoter, transcribed, bivalent, distant, proximal, heterochromatic, polycomb, poised, many different types of things, quiescent, transcribed, weakly transcribed. Yeah. Transcribed early in the gene, transcribed later in the gene. They all have actually different combinations of marks. Yeah. So what you're asking is whether an enhancer state and a promoter state are on top of each other? Yeah, so it's a very, very important question. And we're going to talk about the posterior probability of state assignment for a particular region. And in some cases, it will be ambiguous. We won't quite tell what state it is. And those places might correspond to something that's sort of in between a promoter and an enhancer, or a transcribed enhancers and so forth. And we'll basically have the entire machinery that we've learned about with hidden Markov models already at our disposal. So we can do posterior decoding, we can do Viterbi decoding, we can look at the actual, posterior probability of every state, look at the second best state, look at alternative parses and all of that. You are the best. I think one box is plenty. Yeah. Let's see now if they'll take it away. I'll take the big one. So I can put my mouse next. MIT, the latest technology in classroom. This is awesome. So my father always tells me, when I was a student, I would go home and we would have one big room for all of my siblings and I would work on a cardboard box to do my homework. I can tell my dad, and when I taught at MIT, I used a cardboard box as my podium. So anyway, this actually works great. I have kids and I bring them toys and they'll open the toy, they'll open the box, throw away the toy, and start playing with the box. They'll just make it a train and then a plane and then a transmogrifier and all kinds of stuff. So cardboard boxes are awesome. All right, any questions so far on life or Epigenomes? Yeah. Are you asking about a specific region example, or are you asking, do they really change the function of the underlying region? It changes how things are interpreted. It changes what machinery will be able to access the DNA. And that in turn changes what will be transcribed. So if a promoter is in a closed chromatin region, RNA polymerase will not be able to bind there. It will sort of try and fall off. If the promoter is accessible, RNA polymerase will be able to come in and start transcribing. So whether a particular gene is transcribed or not depends on the chromatin state. But the chromatin state establishes many things. It can establish, for example, whether their state will be poised for activation. After transcription starts, polymerase can actually stop there and pause. That pausing is also encoded epigenomically through specific combinations of marks. Whether a particular region was previously active and has now been repressed is encoded in the epigenome rather than a region that recently became active without previously being actively repressed, being quiescent. So there's a lot more information than just on-off, if that's your question. No. So these are general factors. So these histones are everywhere in the genome. And the specific epigenomic modifications that they have mark classes of regions, not specific regions. It won't tell you, hey, here you are on chromosome 13, position 247,000. No, that's not encoded in the epigenome. But what's encoded in the epigenome is, hey, here's all the active regions. So I like to call it data-centric indexing rather than address-centric indexing. So the way that early computers worked were go-to statements that basically said, go to that point in your memory and then start executing here. The genome does not work that way. It doesn't know where it is. All it knows is, all the regions that need to be turned on in neurons, put on a yellow flag. And then the same proteins will land in the yellow flags. And then in the liver, the yellow flags will be in other places, but the same machinery will land in the yellow flag. Does that answer your question? We will talk about that. So we will talk exactly about all of that. So we'll talk about how to discover these combinations in an automated fashion. We'll talk about how to assign functions to these combinations in automated fashion. Great. Okay, so basically, we are now able to map these modifications across many different cell types. So this is one project that my group was very heavily involved in, the Roadmap Epigenomics project, where we basically mapped across an international consortium of collaborators all of these different tissues for many of these different modifications. So we mapped, promoter-associated marks, enhancer-associated marks, transcribed marks, polycomb marks, heterochromatin marks, activity marks, accessibility marks, methylation, and transcription in each of adult tissues, embryonic tissues, iPS lines, embryonic stem cells, in vitro differentiated lines that basically tell you about different tissues and what they look like. And then this was done across many different projects. So the first one among them was the ENCODE project, the Encyclopedia of DNA Elements. That started out on 1% of the human genome. The group basically said, hey, let's just study 1% and just do all these experiments on this 1%. And then new technologies came about for being able to sequence rather than hybridize. So for the 1%, the project was basically building tiling arrays of 1% of the human genome and then hybridizing things through these arrays. And then next generation sequencing came along and ENCODE project basically scaled up to the entire genome. The Roadmap Epigenomics project basically sort of worked in parallel with ENCODE and then became the International Human Epigenome Consortium together. The Blueprint project in Europe basically sought to do the same exact thing, but across many blood cell types and across more individuals. Mouse encode for mouse, and then mod encode for model organisms, Drosophila and worm. And for each of these projects, they seek to map multiple modifications in multiple cell types, in multiple individuals, in multiple species, in multiple conditions, in multiple antibodies, and across the whole genome each time, basically requiring this massive, massive infrastructure of tools that I'm going to talk about today. So there's many waves of papers that have already been published. There's many more that are in the pipeline, and now the time is right for massive, massive analysis. So now's a good time to be an epigenomicist. So this is, the introduction, basically what is epigenomics good for, diversity of modifications, and then let's now learn about, that should have been here, ChIP-Seq. So how do we map these modifications? So with bisulfite, with DNA methylation, we basically use bisulfite sequencing. So unfortunately, I don't have a slide for that, but you can transform the DNA in a way that distinguishes between CpG dinucleotides and that are methylated and those that are not. And you can then sequence the genome twice, once after modifying it, once without modifying it, and then the difference will basically tell you which positions were actually DNA methylated. Everybody cool with that? So for DNA accessibility, what you can do is chop up the DNA based on a protein that will digest open chromatin regions, and then you just sequence the ends of the fragments that were digested, and then that will tell you where the open chromatin regions were. Who's with me on this one? Great, awesome. So that's for DNA methylation and for DNA accessibility. What about for histone modifications, where they're much more complex? You have to actually figure out where a particular modification sits across the entire genome. How do you do that? And that's, I think, related to your question. You basically do that by making a bunch of that modification and then feeding it to a cute little bunny. And then the bunny will make a bunch of little antibodies. The immune system of the bunny will react to that foreign object. And then you will actually have the serum of the bunny that will actually have a lot of that antibody. And then you can use that antibody, you can couple it with beads and pull down regions of the genome that this antibody now recognizes. And these regions are the ones that have the modification that you fed to the bunny in the first place. You can use all kinds of really cute animals, you can use camels, you can use goats, all kinds of things. Okay? Anything with an immune system. Who's with me so far? So once you've built your antibody, you basically are ready to pull things down. And how do you pull things down? You basically do a step known as cross-linking. That basically freezes proteins together with DNA so that they're stuck. And then you chop up your DNA, and then you basically pull down all of the regions that your antibody recognizes. You reverse the cross-linking, you throw away all the protein, and you're left with a bunch of DNA segments that were pulled down based on your antibody. What do you do then? You can then sequence, or you can hybridize to a microarray. Okay? So the whole concept is that I want to figure out where was a particular antibody or where was a particular transcription factor. I'm going to build antibodies against that transcription factor or antibodies against that modification. I'm going to then pull down all regions that have that modification, get rid of the protein, only keep the DNA, and then sequence the DNA. Raise your hands if you're 100% with me. Awesome, love it. Good. So what you end up with is sequencing reads, a lot of sequencing reads. And what you need to do is then map them systematically to the genome and then figure out where every modification was. Okay? And this is what the mapping looks like. You map these hundreds of thousands of reads, actually tens of millions of reads, onto the genome, and you end up with a profile that basically tells you, ooh, there's a bunch of reads that mapped here, fewer that mapped here, and more that mapped here, and so on and so forth. Everybody with me on this? And that basically tells you that this region here had H3K4 monomethylation. This region here had H3K4 trimethylation. Notice that these are the enhancers of that gene. That gene is transcribed from the right to the left. You can see the little arrows moving to the left here. And then the enhancers are flanking the gene, and a lot of them are in the first intron of the gene. Then the promoter marks are closer to where the promoter is, right where the transcription starts. And then H3K36 trimethylation is marking the gene body. Okay? Who thinks this is kind of cool? Right? We can just read off what's encoded in the DNA without even looking at the DNA. We just look at the epigenomic marks, and that actually gives us an information of what's actually encoded in there. And that's how the genome knows, to be honest. Every cell knows what's important, what's transcribed, and what's, what are the, the body of knowledge of that cell among the human encyclopedia based on the regions that are marked. Yes. Correct. Correct. Well, you're so fixated on transcription. Transcription happens much, much later on. The epigenome basically encodes this diversity of functions, one of which enables genes to be transcribed. But basically, for now, I'm only focusing on sort of what is the landscape that the transcription machinery will see as it's approaching this gene. And it will basically say, ooh, enhancers, I like that. So all these, proteins that are bodies of RNA polymers will basically show up, and they'll start having a party on the enhancers. And then they'll be like, polymerase, want to join us? And polymers will be like, well, I need some promoter marks. Like, we've got them for you. And polymers will basically show up and start attaching here. And then a bunch of additional factors will come in and initiate the process of transcription. And then transcription will basically start happening. And perhaps along with transcription, there will be other modifications that are established on the body of the gene itself that will then tell us that this region has been transcribed. Does that help? Awesome. All right. So basically now, this is what it looks like for 3 marks. This is what it looks like for dozens of marks. The big question is, computationally, how do you map 20 million reads to the genome? And I alluded to that at the last lecture when we were talking about RNA sequencing, sort of mapping these short reads very fast. And I told you that, well, the traditional hashing schemes basically create this massive, massive hash table for the entire genome. And then they go through this hash table, assigning every read at a time by doing all these steps of sort of looking at where it goes, aligning it, finding a hit, a seed, extension, chopping up, scoring, and so on and so forth. So that's very, very fast. That's ridiculously fast compared to the traditional alignment that we saw that basically takes order n squared. These hashing schemes are linear and they're super, super fast. But they take a lot of memory, which basically means that in practice, they actually run slower than they could if they used less memory. So that's what the algorithm that we're going to see today addresses, the Burrows-Wheeler transform. So what does it do? It basically maps reads to the genome. It aligns reads to the best matching location in the reference genome. And usually you have tens of millions of reads, each of which are 30 bases long. And here's one example. And additional complications is that you may want to allow mismatches to deal with sequencing errors or to deal with genetic variants. And what you need is both algorithmic efficiency and memory efficiency. Okay? And all of you probably have already found this string. Anybody wants to just raise their hand? Where is it? Anyway, this is a tiny, tiny little subset of the genome. Try doing this over 3 billion bases. Okay? Big, big genome. So how do we do that ridiculously fast? So again, you could do sequence alignment, you could do hashing, you could do linear time string matching, you could do suffix trees or suffix arrays. The problem is that a lot of them are memory heavy. And today we're going to look at something that actually has a very, very small memory footprint. Okay? And this basically became the new norm since 2009. If you look at 2009, basically this paper appeared by Ben Lagmead. This now has thousands of citations. And this introduced, remember, the tuxedo suite started with bow tie, BWT. It was just a cute name for something that had BWT in its name. And it was 30 times faster and 300 times faster than previous algorithms. Okay, so that made a big, big difference. And then after 2009, every single program came up with version 2 that at the core had Burrows-Wheeler transform. Okay, so this was, sorry, pun not intended, transformative. So let's compare hashing to Burrows-Wheeler transform, BWT. So hashing basically, takes your reference genome, creates this big, big, big table, and then every time you have a short read, you basically break it up and you look at what are each of the components matching inside your big hash table, and then that tells you where it goes in the genome. Everybody with me so far on the left? What Burrows-Wheeler transform does is that it concatenates all your chromosomes, and then it shuffles them in an interesting fashion that basically creates a modified version of the genome, which is still linear, and it's still the same length. It's 3 billion bases long. This transformation was initially developed for compression. If you look at bzip2, for example, in your Unix system, it actually uses the Burrows-Wheeler transform to compress. So why would anybody in the right mind use this for searching? Well, turns out that it has some very, very cool properties whereby strings of characters that are identical are actually, matching near each other. And when you search for a small string, you can actually start growing the string from the right to the left, looking at increasingly larger suffixes of whatever string you're searching. And this BWT transform has the very, very cool property that if you start with all suffixes of, that start with a T, they will all be here. And if you look at all suffixes that start with an AT, they will all be here. And those that start with AAT, they will all be here. So what you're doing is that you have a laser beam that gets progressively narrower while searching increasingly long substrings of a particular string that you're trying to find. Okay? Once you found all those, you basically, every time you search a character, you just do a bunch of pointer games. This is your first pointer, this is your last pointer. You just go tack, 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 tack. All you're doing is, sort of moving these pointers around. Extremely, extremely efficient operations, no moving data around. And then you end up with exactly the location where all of the matches for this particular string are found. And if you have many such locations, they'll all be consecutive. They'll be back to back from each other. Okay? And then you basically can pile up your reads in this transformed genome. Once you have piled up all your reads, you can retransform it back exactly once. And then, boom, you have all your counts for all your reads in the original genome. Except for the magic for how this happens, who's with me on what we're trying to achieve? Raise your hands. Awesome. Great. So let's now see, how do we make such a data structure that has this very, very cool property? Okay? Here's BWT in one slide. So what we're going to do is, sounds a little crazy, but we're going to transform the word banana into bna. Okay? Don't take this out of context. But the function BWT is going to basically create a table whose rows are all possible rotations of our original string. It's going to sort all of the rows alphabetically, and it's going to return the last column of the table. You're out of your mind. So here's all rotations of the word banana. It starts with a funny character that's only at the beginning, funny character that's only at the end, kind of like bananas. And then you just sort all of that alphanumerically, and then you get the last column. Okay? Everybody clear as to not why we're doing it? This makes no sense. But what are we doing? Everybody clear with what are we doing? Raise your hand if you're clear with what are we doing. Awesome. Good. First, I'm going to tell you that whatever we did to mess up our genome is reversible. And if you give me, banana, I can just recover banana in a series of little steps. Okay? Who sees already how it's reversible? Awesome. One person sees it, two people see it. That's cool. So I can compute the inverse BWT using the following process. So if I have the last column, how do I recover the first column? Remember, the first column is actually super cool. It's ABN. Why is that super cool? Because it is. Say it again. It's in alphabetical order, right? Notice, if I do a bunch of rotations, every column has exactly the same characters in a different order. So now, all together now, how do I get the first column if I have the last column? Sorted. Is that super cool? Awesome. So I can sort the last column to get the first column. Now, how do I get the second column? And then boom, you have it. Notice, every row has exactly the same characters in exactly the same order. They're just rotations of each other. Right? I can read banana starting at any position in a different row. So if I know a character in a particular row, I know the character in the row next to it. In the same row next to it. Okay? Because it just reads banana. Got it? Awesome. Anyone else has it? So now, if I have the last column and the first column, what do they have that's special? If I have the last column and the first column, that means I have every doublet. Why? Because I can read BA here, and I can read NA there, and NA there, and so on and so forth. Who can see this? How, because of the rotational property, if I have the first column and the last column, I can just put the first column next to the last column, and I will have all pairs. Who's with me on this one? Raise your hands. Some shy hands. Okay? So that basically means that if I have all doublets, what do I need to do to get the first two columns? All together. Sort them. Right? So I have the last column here. I sort it. I add the first column. Basically, I then sort the doublets, and then I can just add that to the last column. And I can sort it again. I can add that to the last column and sort it again, add that to the last column and sort it again. Everybody with me? And then when I'm done with the whole process, I basically have the full matrix. And I get my word wherever these funny start characters. Who thinks this is like ridiculously cool? Raise your hand. Awesome. I think it's ridiculously cool. I still haven't told you why this is useful. I just told you that it's possible and it's already super cool. Let's see how it's useful now. Everybody with me, right? We can transform and we can reverse transform. Okay? And then I can actually compress this because it has this property that, strings of characters are close together. Yeah, it sort of depends on your nomenclature of where these characters should be. But yeah, I agree with you that we should decide whether the start character should always come first and the end character should always come last. But it's invariant as to whichever way you define that this should sort. As long as you're consistent. All right, good. So now let's search for an exact match. Remember this property here of my beginning and end of every substring is going to be this narrower and narrower and narrower laser beam. And at the end, I'm going to end up with exactly the position where all of the matches are going to be. All right. So now, if I have my entire matrix, it turns out I don't need the entire matrix. I just need the first and the last column. And the first column I have already, that's my genome. And the last column is basically the BWT transform. Okay. So if I have basically the first column and the last column, in fact, I don't even need the first column and the last column. All I need is 2 pointers that basically tell me where's the beginning of the laser beam and where's the end of the laser beam. All right. So I'm not so self-centered that I made this slide myself. This was made by Jason Ernst, who guest lectured this lecture. So I'm kind of stuck with my name on the slide. But this will eventually become Jason Ernst. He doesn't have enough repetitive words in his letters in his name, I guess. But anyway, Manolis Kellis is the string here, and then here's all the permutations of that, and then you can then sort it and then get the last column. Okay. So if you're searching for the string OLIS in this genome, how are you going to find it? Well, as I mentioned earlier, you start from the last character and you progressively include more and more characters. And the last character has a very wide laser beam, which is getting narrower and narrower and narrower. All you need to know is that S is so for every letter, you basically know where's the first position where it appears. And, letter A first appears in position 2, letter E first appears in position 3, and so on and so forth. And then I basically go down and find wherever S appears and just how many S's are there. And that gives me the width of the first laser beam. And then I know that as I add one more character, it's going to be here because all of these are rotations of each other. So I'm just going to search here for the characters I and where they go. So I can just look up I, and it starts here at position 4. This was fixed in the previous version a few moments ago, but anyway, I starts at 4. Sorry. This is already fixed somewhere. And then from position 4, it's length 2, so I'm here. And then I add one more character. I'm increasing my suffix with increasingly long stretches. And every single time I'm searching for that prefix in the sequence. And then that basically tells me L. And where is L? L is at position 6, and it has a scope of 2. And then I'm looking at the end. Now, finally, I have a mismatch. So that beam is going to get narrower. But all of the O's are going to be together. And then I go to the O, and it's length 1, and boom, I get my OLIS. Okay? Yeah. Oh, that's just a substring that you're searching. You just did, because by getting, basically you're searching a substring. This is your genome, and you're searching for the substring OLIS. But you've already looked at all of the places that end in S, and then IS, and then LIS, and then OLS. It's the prefixes of my suffix that I'm searching. So that's why when I end up with here, when I end up at this O, I know that everything that follows is already matching my increasingly large search string. Yes. You can basically use the properties of this table to actually not have to search every one of them. Because of the way that they're sorted, you can actually, look it up directly. You don't have to actually search. So, you're playing with these pointers of how long is each run. And then you know that this run of O, for example, is exactly 1 character long. You don't have to search the entire thing. You can only search for that. Exactly, exactly. Basically, you know, in this particular case, there's only one O in this genome, but yes, it's going to be only those that have the same suffix of what you've already searched. And that's how that beam is getting narrowed right now. Ready with me? So that's the algorithm. That's the Burrows-Wheeler transform algorithm. That's why it's useful, basically, because you do this transformation once, it's extremely memory efficient, and then when you're searching for increasingly long substrings, you can basically narrow down that beam with only playing with 2 pointers, the start and the length of each run. And by sort of jumping back and forth between the first and the last column, you can effectively do that very, very rapidly. Who's with me so far? Raise your hands. Awesome. Great question. Mm-hmm. No, you need both. I mean, it's helpful to have both, but you're right. You could, I mean, it makes very little difference implementation-wise. Yeah, but these special characters are always helpful in sort of knowing where you are and whether you ended. It's probably helpful in knowing that you don't have full rotation. You have to stop there, that your word actually ends. Yes? Correct, correct. And you basically do this transformation after you have a vector of count for every location in the genome. You transform all of these counts once. So the expensive operation of this transformation, you only do it once. All right, good. So now I've basically mapped everything. And the key advantage is that there's very little memory usage. It's the same as the input and sometimes less. You don't represent the matrix or strings. You only keep pointers. And you encode by simply sorting the pointers. And you decode by simply following the pointers. As I mentioned, the original application was string compression, where the runs of letters were compressed into letter run length pairs. And for bioinformatics, it's used for substring searching. And it achieves similar runtime as hash tables and suffix trees, but it's extremely memory efficient. So in practice, it is hundreds of times faster when you actually run it on the machine. And if you map hundreds of thousands of reads, you only transform once, you pre-process once, you read the count in the transform space, and then you reverse transform once, and you map the counts to the. Okay? All right, so we've now mapped all our reads. We've basically done ChIP-Seq. We've mapped all our reads using different methods, and now we'd like to do some quality control. We'd like to know, is this a good experiment? So ENCODE developed some uniform processing pipelines that have become quite standard in the field since then. So, you first map the reads, and then you call peaks, and then you search for whether the peaks are actually reproducible, and then you do some motif discovery to look for, common sequences in the peak. Then you do expression analysis or co-association analysis. You can look at the aggregation of the signal. And you can actually segment the genome using this Chromatin state model that we're going to talk about and learn about the co-occurrence status of different segmentation. So the first quality control, and basically all of these rely on being able to actually carry out the different quality controls which happen at different stages in this pipeline. So the first quality control is basically using input DNA as a control data set. So I mentioned earlier that you basically build these antibodies against the particular transcription factor that you're interested in, and you pull them out of the genome. The problem is that if you carry out this approach without an antibody, you're going to end up with something like this. It's not just uniform background, it's got peaks, and those peaks are indicative of properties of the genome that are very different from location to location. So the way that you usually look at these chromatin immunoprecipitation experiments, these ChIP-Seq experiments, is by basically doing your ChIP-Seq experiment twice, once with your antibody, once with your naked control, basically. And then in both cases, you pull down chromatin, and then you do the mapping, and you take the ratio between the two. Okay? Everybody with me on this one? The second one is basically asking, are the reads that I'm obtaining high quality? And, you know, this is simply looking at the quality score of the bases, and that's based on whatever comes out of the sequencing machine. That basically tells you, yes, the signal intensity between A, C, G, and T was in fact strong enough that I can distinguish the bases. And if you can't, you just throw these away. Okay? The third quality control is what fraction of your reads you can actually map to the genome. So that basically is reflecting of, you know, if it's just a garbage experiment where basically, you know, a bunch of DNA came over from random places. And then, you know, whether these reads map uniquely to the genome or whether they map multiply to the genome or whether they don't map at all to the genome is basically very indicative of, you know, your overall quality of the experiment. In a very good experiment, most of the reads, in fact, map uniquely, and a handful map in, or some map in repetitive places, but then you can identify those because they are in repetitive places. The next quality control metric is about your library complexity. That basically tells you just how many reads, how many DNA fragments were there to start with. Because if I do my chromatin immunoprecipitation and I end up with very little DNA, say I end up with 10 fragments, it doesn't matter if I sequence 10 million reads. All these reads are going to come from the same 10 fragments. Okay? And that's basically telling me that I just didn't pull down enough DNA, enough sort of unique regions in my genome. So what you can look at to know if your library has sufficient complexity is the start positions and the end positions of the reads that you're actually making. And if they pile up on top of each other without capturing the surrounding spaces, it basically means that your DNA was simply, your library did not have enough complexity, did not have enough unique locations. So it doesn't matter if you sequence another 10 million reads, you're not going to get any new locations. What you want your library to look like is this, basically just random start and random end. Okay? And you can do that by basically asking, what is the number of locations that map exactly once? What is the number of locations that map exactly twice? And, you know, taking the ratio between those. And, you know, if that ends up being much higher, then that's a problem. So basically, uniquely mapping versus double mapping should be a very high fraction. Everybody with me on this one? So so that's at the level of reads. You can also do an analysis on the relationship between the reads and how they map on the genome. And here's the key idea here. If you have some transcription factor, some nucleosome that's sort of sitting here in the genome, the fragments that come down with this nucleosome are the fragments that were actually attached to it. And you will not attach to a fragment that stops halfway. You have to attach to a fragment that actually traverses this, right? So if I have blue reads that are sequencing towards the right, or red reads that are sequencing towards the left, the blue reads better start, you know, to the left of this point, and the red reads better start to the right of that point. Okay? Is everybody with me on that? So that means that the true distribution of where that protein or that nucleosome was is in fact exactly halfway between two separate peaks, one peak coming from the forward strand and one peak coming from the reverse strand. So all the reads that I'm sequencing from the left to the right are going to be mapping here. All the reads that I'm sequencing from the right to the left are going to be mapping here. Who's with me on this? Raise your hands if you are. Awesome, great. So that basically means that after I do all of this, I can then recognize two distinct peaks. And that basically means that I can actually start asking, what is the correlation between the reads that are coming from the left or the right? And that's what strand cross-correlation analysis does. It basically says, where are all the reads that are coming from the forward, all the reads that are coming from the reverse strand? And is there a characteristic distance between them that indicates what is my fragment length distribution? Because these are the fragments that are going to be spanning wherever that protein or that nucleosome was. And as I shift the reads past each other, at exactly halfway between these two fragment lengths, these two ends, I will get a maximal correlation. So at offset A, I get a little correlation. At offset B, I get more correlation. At offset C, I get maximal correlation. At offset D, I get low correlation. Okay? And the shape of these cross-correlation analysis, again, tells you a lot about the quality of your experiment. Who's with me on this one? Raise your hands. Awesome. Great. Love it. So that's the length at which the ChIP-Seq DNA was in fact fragmented. And these fragments are basically, you know, exactly. Exactly. And that's because very long fragments can actually exist. As long as those fragments are capturing your protein, you're going to get a longer tail towards the outside. And that's the tail towards the inside. So the inside is always narrower, the outside is always longer. Just because of the nature of the fragmentation, basically. So longer fragments are here, which are spaced, you know, far from each other, just because of the way that you fragment the DNA. If you're fragmenting, because the way that you cut things, there's a log property for how the DNA flows. When you cut two ends, one, you know, at the same location, one of them is going to be narrower and the other one's going to be longer. So anyway, you basically have your fragmentation length, and you also have your read length. If you have your read length, that is an artifact of reverse transcriptase going for, or no, this is your, anyway, to sequence DNA, you need to go from single-stranded DNA to double-stranded DNA, and that's your primer, and basically restart on the opposite side of your read. So just as you're done making a read, if you restart and you sort of make that complement, then you're going to end up with fragments that are a read length apart. And some really, really bad data sets look like this, where you basically have most of your pairs coming from that fragment, from that read length rather than the fragment length. The sort of good data sets look like that, where this is almost negligible. And then, you know, you can have anything in between. Okay? So anyway, these are cool ways that you can sort of use to sort of understand the properties of whatever comes out of the sequence. Now, the next step is to go from this continuous signal on the DNA to actually intervals where you think that a particular region is bound. And this is largely a ill-defined problem, because the way that you define these intervals takes so many different parameters that you could use. But you know, there's been a huge number of peak callers that have been developed. Max and ChIP-Seq were the ones that are very heavily used in ENCODE. They're also very popular, even outside ENCODE. Max is, you know, particularly widely used. It was developed by Shirley Liu over at Harvard. And, you know, these basically rely on a model for how reads are distributed. And they basically assume some kind of Poisson distribution of reads from the genome. And then you can adjust that locally and call peaks when this Poisson distribution exceeds a certain threshold, which indicates that reads were probably sampled from that location at some higher frequency. And then you can calculate that frequency. So the problem is that as you sequence experiments at different depths, they can actually lead to different thresholds for peak calling. And those thresholds can actually have a very big effect on reproducibility. For example, if I sequence one experiment more heavily than the other experiment, you know, one is going to have 100,000 peaks, the other one is going to have 20,000 peaks. If I want to require that peaks are agreeing between the two replicates, then I'm going to end up with 20,000 peaks. Every single time I'm going to be constrained by the worst experiment, the worst replicate that I did, rather than utilizing the best replicate. If I take the union, the problem is that it's as if I didn't do replicates, as if I sequenced more deeply one experiment. So how do you deal with this problem? You basically have many, many peaks in one experiment, few peaks in the other experiment. How do you leverage the fact that they're truly replicates? Well, what you can do is sort all your peaks based on their intensity. And you can keep down the intensity rank of the peaks in your deep experiment until you stop replicating in the shallow experiment, okay? And this is the idea. The idea is that true peaks will be highly ranked in both replicates. So you can just keep going down the rank list until the ranks are no longer correlated. And then the cutoff will be different for the two replicates. If I go down my very good experiment, and then I notice at some point my peaks stop replicating, that basically means I should stop here. But here I stopped at 50,000 peaks, even though only 10% of them were captured by my weaker experiment. I have 50,000 peaks in one experiment, only 5,000 peaks in the other experiment, and yet I can use the less good experiment as a replicate for my much better experiment, okay? We're basically looking at the rank correlations and where they start being non-correlated. Who's with me so far on this one? Raise your hands. Awesome. Great. All right, so that's basically, you know, what IDR does. And then you can actually do some funky tricks to use pseudo replicates rather than just replicates. And you know, you can basically resample from the same experiment. All right, so that's all at the level of primary data processing. So basically, I've mapped my reads to the genome, I've called peaks, I've decided how many peaks to keep, I've basically used various quality control metrics, cross-correlation, you know, and then this IDR, this irreproducible discovery rate. So just like the false discovery rate, this is an irreproducible discovery rate, and it's about reproducibility rather than truth. Okay. So now we'd like to go a step further and start interpreting the data at a higher level. And that's basically trying to understand, how do I define different classes of epigenomic elements based on the combinations of marks that are there? And as I've mentioned before, a very natural way of doing that is to develop a hidden Markov model. And a hidden Markov model will basically have the hidden states of the chromatin that most likely gave rise to the observed histone-modification marks. And this is not just a hidden Markov model, it's a multi-variate hidden Markov model, which basically means that every state emits not just one variable, but many variables. And therefore, from every state, I have a whole vector of histone modification marks and the probabilities with which each mark will be, emitted. So then the key design choice that was made in this particular instantiation of hidden Markov models called Chrome-HMM. This was developed by Jason Ernst, a postdoc in my lab, who's now a professor at tenure professor at the University of California, Los Angeles, UCLA. So the idea there is that, you know, what we'd like to do is truly understand the combinatorics. And therefore, the big design choice that we made was to not model the signal intensity directly, but to instead locally binarize the data. And that basically means that for every location, we get a 1-0 presence-absence of every mark. And the way to do that is to basically, again, develop a Poisson random variable that basically tells us for every location, there's a probability of obtaining something in that location. And then when that probability exceeds some threshold, it basically says it's really unlikely that the signal here comes from random noise, then you basically make that a 1. And you adjust that threshold based on your sequencing depth for every mark independent. Once you have that, you can basically learn an emission matrix using Baum-Welch and a transition matrix, again using Baum-Welch. So this is the expectation maximization unsupervised learning algorithm that we let loose on a set of 41 genome-wide histone modification tracks. And then it came back with 51 different chromatin states that capture different combinations of these marks. In this particular set of states, you have, you know, all of these different acetylation marks, and then all these methylation marks. In these other states, you have all of these, you know, transcription-associated marks, cancer-associated marks, promoter-associated marks, and so forth. But the hidden marker model doesn't know anything about that. It is trained in a completely unbiased way, without any kind of annotations, and without knowing that enhancers and promoters even exist. So what it basically learns is combinations of marks that are appearing together more often than you would expect by chance. It partitions effectively the genome into segments that have more uniformity than what you would expect by chance. And then it also learns the spatial relationships between neighboring states, therefore revealing distinct subgroups of states based on the transitions between. So what we're capturing is, on one hand, the combinations of marks on the genome, and on the other hand, the spatial relationship of these biological functions of the genome. So we let the algorithm loose on the genome, and it came back with a particular annotation that you can see here. So this is a particular gene in the direction of transcription. And then the algorithm said, oh, there's these, states here, which we later called promoter states. And then these early transcription, middle transcription, late transcription states, very different chromatin marks for the different positions. And there's these active intergenic states that have these enhancer characteristics flanking the transcribed regions. You have repressed regions further on, and then repetitive regions even further on. The complexity of the model matched the complexity of the genome. That basically means that more states were assigned to tiny little fractions of the genome, and then a small number of states covered the vast majority of the genome. That basically means that it didn't just partition the genome in equal size bins. It said, well, there's these promoter states that are extremely important, even though they only cover 1% of the genome. And this is sort of the genome-wide classification of all 23 chromosome pairs from beginning to the end. So you can see here some of these centromeres or heterochromatic regions are appearing, very, very distinctly. You see these gene-dense regions lighting up in red. You can see, all kinds of features that are immediately visible. So the next challenge is great. We let that HMM loose. It learned a bunch of chromatin states. How do we annotate the states? How do we know what they mean? So we go from the state definitions to names and properties for these states. And the way we do that is by looking at existing annotations on the genome and then using them to annotate each of the chromatin states. So for example, we can ask, what fraction of the genome do they cover? What percentage of that chromatin state falls within 2,000 base pairs of the transcription start site? And you find that this entire group of states, which we later called promoter states, are in fact very heavily enriched here. What fraction of them overlap genes, gene bodies? And we found that all of these states, which we later called transcribed states, are in fact overlapping genic regions. You can ask what fraction of them overlap lamina-associated regions, which is the nuclear periphery that we're going to talk about on Thursday. And we found that all of these states, which we later called repressed states, are in fact very strongly enriched for lamina. Then we asked what fraction of those overlap repetitive states, repetitive regions of the genome, repeat elements. And we found that all of these states, which we later called repetitive states, are in fact overlapping those. So what our algorithm was doing is partitioning the genome into different classes that had dramatically different properties. So that basically means that simply from the combination of chromatin marks in a completely unsupervised way, emerged a partitioning of the genome that made complete biological sense. Who thinks this is kind of cool? This is exactly the hidden marker model that you learned before. Just let it loose on the genome, and it learned all these things. So then we used all of that information to then annotate every state with, quite detailed nomenclature. And here they are. Okay? We found all kinds of really cool things. We found these ZNF repressed states, which is still one of my favorites, that had 112-fold enrichment for zinc finger protein being encoded there. This is very weird, right? Like, there's a class of genes that have their own chromatin state. We found a particular state for transcription end, was not previously known to exist. We found that different classes of genes are enriched for different promoter states. That there's something encoded in the epigenomic landscape that is in fact reflected of the type of function that is encoded there. We found that different transcription factors enrich for, different classes of promoter states. And similarly, different motifs mark enhancers versus promoters. And, different repeat elements are associated with different genomic markings. We also use that to define new protein coding genes that were previously missed, because from their epigenomic signature, we could recognize them. We use that to define 2,000 different long non-coding RNAs that showed all of the signatures of promoter, transcribed, but didn't show any protein coding constraint. We found, new developmental enhancer regions. We found candidate regions from genetic association studies that we can now annotate as putative regulatory regions. And we basically, discovered that we can, in fact, recover promoter regions and transcribed regions with very, very high accuracy. So a lot of people have asked me, how do you select the number of states? So this is actually a very good example for how to deal with model complexity. So the idea here is that we have a multi-variate hit marker model that emits vectors of marks rather than individual marks. So what we can ask is, for every pair of chromatin marks, is it dependent or independent? And does it become conditionally independent based on the set of chromatin states? So what we're going to do is ask if they are truly independent, then the probability of them occurring together will simply be the product of each of their individual frequencies. But if they are dependent, then we will see a skew away from the diagonal. And what was very cool is that as you increase the number of states, these marks became more and more independent of each other. So that basically means that our additional states were capturing some of the mark co-occurrence properties, which were now becoming conditionally independent, given the chromatin state. Okay? So that's one way to basically understand that the increasing number of states actually captures a lot of these mark dependencies. But where do we stop? How do we know that we have enough chromatin states? And you can use something as a Bayesian information criterion, which basically tells you how much do I penalize my model complexity versus how much more accuracy do I get in modeling my genome. And what you can see is that this simply keeps rising. Doesn't matter how many states you give it, it'll just want more states. There's no such thing as a optimal number of states. But what you can do instead is first learn a larger model that captures, presumably all the relevant states, and then prune that model down by greedily eliminating the least informative states, and then select some cutoff based on where you want to stop. But what's really interesting here is that given the, say, 51 chromatin states that I end at here, these 51 chromatin states are much more informative than if I had just selected the first 51 states that are rows. So basically, if I had random initialization, I wouldn't be able to sort of capture all these chromatin states, but given this model pruning, then I'm able to actually capture them much more interesting. Okay. So that's about selecting the number of states. But up until now, I've only told you about multiple marks in one cell type. What if I have many cell types, which is typically the case? I mean, I showed you this example where we had 127 different tissue types. So if I now map 9 marks in 9 cell types, what kind of model do I learn? These basically become 81 different chromatin mark tracks. So what I could do is simply learn a single model with 81 emissions. Basically, every state emits a vector of 81 different tracks. However, they're not independent of each other. I mean, they're very highly coupled. They're the same marks in different cell types. So the way to do that is either to learn independent models in every cell type, You basically learn, say a 15-state model in every one of your cell types, and then you just cluster together and just hope that you've learned the same. The alternative is to stack your cell types together and just learn one very big model. And the third approach, which is the one that we used, was to basically simply concatenate the genome. Instead of just stacking the chromatin, the cell types on top of each other, you basically just concatenated chromosomes next to each other. So instead of having 23 pairs of chromosomes, I had 23 times 9 pairs of chromosomes. That allows me to now, in the same way that I have chromosome 1 next to chromosome 2, etc., just have chromosome 1 from the other cell type, chromosome 1 from the other cell type, and so forth, which basically forced the model to learn the same state definitions in every cell type. And that's sort of going back to the question that we had earlier about, are these marks having different meaning in every cell type? What we found is that if you relearn the same model independently in each of the cell type, and if you learn it jointly by concatenation, you end up with recovering the same set of chromatin states. And that's something that we just did not know what to expect when we first did. The challenge, of course, is that you may have some missing experiments. So basically, in one cell type, you may be missing those 2 marks. In another cell type, you may be missing 2 other marks, and so on and so forth. So how do you deal with that? Well, you could basically let your model treat the missing tracks as missing data. And hidden Markov models have the entire machinery that you need for that. By basically integrating over the missing data. It's more expensive computationally, but you can do that. The other option is to simply say, well, if I have these and these data sets, I can use the correlation patterns between cell types and between tracks, between different marks, to basically impute the missing data. And that is what we did. And I'm going to skip some slides here. A lot of slides here. So you can actually, we'll cover them in a regulatory genomics lecture. So what we did actually, again, with Jason, is develop a model that allows us to now impute missing data. And in red here was the data that we had only imputed versions. In blue is the data that we had only observed version. And in purple are the data where we had both observed and imputed. And what we found is that we could actually impute missing data better than the experimentalists could actually generate that data. So every time we compared an imputed data set to an observed data set, to an experimentally assessed data set, the imputed data was actually markedly better using every metric we could think of. And the reason is that every single such imputation track was using information from dozens of other tracks, making it in fact much more robust. So we could use information from other marks in the same tissue by learning their correlation patterns in other cell types and applying them here. And we could use the correlation of the same mark in other tissues by basically learning the correlation properties between tissues using other marks and then applying them here. So that basically meant that for every mark that we wanted to impute in a particular cell type, we could use information from many other marks in the same cell type and from many other cell types for the same mark. And that actually gave rise to, a nearly perfect ability to just predict the data set. You could simply tell us, what data would you like to generate next? And then we would just generate something. You can compare it against the observed data set. And then at every resolution, we found that the imputed data was in fact matching the observed data very accurately. And in places where they did not correlate, it's because it turns out that the experiment itself was a bad experiment. So basically, when we looked at the agreement between observed and imputed, the ones that agreed the least were in fact data sets that were just simply bad data sets, according to many other measures. Okay, so that's where I'll stop and basically say that this gives you an arsenal of tools for dealing with epigenomic data, both for primary data processing as well as sort of higher order processing, understanding model complexity, learning chromatin states, and then imputing missing data. Thank you.